I think we'll go ahead and get started. Well, good morning, everyone, and thank you for joining us for the Great Lakes Seminar Series, which is co-sponsored by the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory. My name is Greg Dick, and I'm pleased to welcome you here today. And it's my pleasure today to introduce our speaker, Amanda Ackiss. So Amanda did her undergrad at the University of Virginia, Wahoo Wah, also my alma mater. And actually, she has an interesting background. Her undergrad was a BA in media studies. So ask her about that later on. She's had an interesting path to where she is now. Let's see. She then did her PhD in ecological sciences at Old Dominion University, and then a postdoc at the USGS Cooperative Fishery Research Unit, which is at the University of Stevens Point in Wisconsin. And then she joined the USGS Great Lakes Science Center here in Ann Arbor in 2020 as a fish biologist. So today she's gonna talk about harnessing genomics to characterize Great Lakes diversity across space and time. And I also wanna mention that we're lucky not just to have her as a speaker today, but also as a co-advisor of a Sigler postdoc, Kristen Behrens, who's in the audience here today. So thanks for visiting, and I'll turn it over to you.
Okay, thanks very much for that introduction, Greg. And it is really my pleasure to been able to make it to the other side of Ann Arbor and actually come into the building. And so in part, I thought I would start my talk before jumping right into the science with a little bit of an overview of who we are on the east side of Ann Arbor as the USGS Great Lakes Science Center. So we are a center that has about 7 facilities that are field stations, biological stations, research facilities, and then we also have a Sheboygan vessel base that's up in the northern part of Michigan in Sheboygan, and we operate 5 large research vessels that are typically relegated to their respective lakes, although periodically they get to jump around and do work on other things. But the headquarters of the Great Lakes Science Center is here in Ann Arbor, and we can actually trace our history in Ann Arbor back about a century. So we can go back all the way to the late 1800s when the U.S. Fish Commission was first developed. The federal government started to be interested in managing their commercial fishery resources that are extremely valuable resources for the country. And we can sort of put our roots at the University of Michigan with a professor known as Jacob Reihard, who was in the Department of Zoology. And he was given federal funding from the Fish Commission to do lake whitefish surveys in Lake St. Clair. So in 1984, those were published. They were really interested, even back at the late 1800s, to understand why lake whitefish populations were declining, because this was a very important fishery species. He did such a great job that as the Fish Commission evolved itself, so in 1903, they became the Bureau of Fisheries within the Department of Commerce and Labor. And those of you who are NOAA affiliated should know maybe a little bit of history. About 10 years later, they split up Commerce and Labor, so it was just the Department of Commerce. But because such great research was being run on Great Lakes fisheries out of the University of Michigan, they kept funding them. So as the Bureau of Fisheries decided to continue giving money to Professor Reihard, he was able to then build out a bunch of PhD programs, including one for one of his students named Walter Keltz, to really try and catalog the diversity of native fishes known as coregonines that were really important commercial and native prey fish in the Great Lakes. And so Walter Keltz here, wearing the very swag hat in the middle there, he ended up publishing a couple of papers about the status of fisheries on coregonines and also sort of the catalog of cisco diversity in the Great Lakes in the 20s. And that was so successful and so appreciated by the federal government that they continued to start funding this work out of Michigan. And so they ended up actually establishing a permanent laboratory at the University of Michigan under another one of Reihard's students, John Van Oosten. So he became the director of the laboratory there, and this is just sort of one of the offshoots of what happened when Van Oosten took over as the director of that laboratory. He hired on a bunch of different scientists, about 5 or so, that were spread out doing a bunch of work, including collecting fish scales. So it's a little small, but you can see it's Department of Commerce Bureau of Fisheries is what we were doing back at that time under the Van Oosten directed laboratory at Ann Arbor. And so they went out, did a bunch of work. And at the time, we have more change to the federal government and where things are aligned. This is where we actually get moved from Department of Commerce to Department of Interior. So 1939, we leave Commerce, we become Interior, and then 1940, we are no longer the Bureau of Fisheries, we are Fish and Wildlife Service. And so continue on with the Great Lakes work that's happening. We end up moving off and having its own federal laboratory built on Green Road that was opened up in May 1996. So this is the first time we actually leave the campus of the University of Michigan, and we are still here today. And then in 1970, NOAA was created by President Nixon's administration. And this is a very unique and interesting situation that a lot of you guys may be familiar with who are working with USGS people, is that they pulled essentially all fisheries, all of the Bureau of Fisheries, then Fish and Wildlife Service out and then into NOAA, with a few key exceptions. One was the freshwater fisheries of the Great Lakes. We stayed in Interior. So we ended up then getting aligned with a U.S. Fish and Wildlife Service. So for a while, we were U.S. FWS. And then in 1994, a couple decades later, we actually were officially renamed the Great Lakes Science Center, and we were realigned with USGS, whose full mission is science production only. So we are not a management-based organization. We don't do sort of management-associated science. We simply do the management-driven science that our partners with FWS or DNRs or DECs or indigenous communities are interested, who are managing these resources. So today, we like to say our science is dunes from the deep. It started with coregonines, it started with fisheries, but we are all over the place now in the science that is driven by management priorities. Lots of invasive species work on things like Phragmites or sea lamprey, but we also do a lot of deep water vessel research as well. So that is where my research falls. And I am sort of still in the umbrella of some of the research that really started the Great Lakes Science Center. I work specifically on a group of fishes known as coregonines that are in the Coregonus artedi species complex. And this is a really interesting complex. This is a group of salmonids that, as the glaciers receded, you started seeing a lot of habitat open up, and they diversified down typically into depth. And so in the large lakes of North America, this is the biggest radiation of salmonids that we see. So Keltz described at least 8 different species when he was funded by the Bureau of Fisheries to go out and try and catalog this diversity. And the peak of that diversity was in Lakes Michigan and Huron. And these are a really critical food source for a lot of the predator species that we're interested in. So walleye, lake trout, things like that. These are sort of their forage base. So once you start having the turn of century come through, a lot of issues were hitting these. A lot of the funding that sort of started our work in this area and on the Great Lakes was driven by a lot of overfishing that was occurring towards the end of the 18th century into the early 19th century. So excessive amounts of overfishing started to have impacts. Things like habitat degradation that would lead to things like sedimentation or nutrient blooms really started having large impacts. But it wasn't until sort of the middle part of the 1900s started seeing some invasive species coming in that really led to sort of the final nail in the coffin of the species complex. So alewife and smelt started getting introduced across the lakes 1950s and really booming in their populations. And they competed in the same spaces that our native prey fishes were existing in. And so they were doing a really good job of outcompeting them. And so we only have a fraction of the historic diversity left in the Great Lakes because of a lot of these impacts. So what we have today is essentially maybe around, or what has been thought around maybe 5 species left in the lakes. So we have a shallow water species that remains in 4 out of the 5 lakes. That's Coregonus artedi, or Cisco. Then we have a deep water species known as bloater or hoyi that's found in 3 of the rest of the Great Lakes. So the upper Great Lakes it's still present in. Another deep water species that's extremely common in Lake Superior. And then we have a couple of species that were thought to exist, but kind of rarely, and mostly really described recently as like periodic one-off catches from Lake Superior. So that's blackfin and or Nigrippidus and Xenithicus or shortjaw cisco. And what you can see is that some cases we had them completely extirpated from lakes like Erie. We had massive reductions in diversity, and where the most intact portfolio remains is in Superior. Now, as the invasive forage fishes have started to stabilize or decline, there's been a growing interest now in managers conserving the native prey fishes that we have. And so in 2018, the Council of Lake Committees endorsed a framework for restoration of native coregonines. I am not going to walk you through this whole slide here. There's sort of 3 components that you could look at it and break it down. Essentially what it is trying to foundation build the science and knowledge in a planning phase, and then undertaking restoration actions, many of which have been under there's a lot of initiatives that have been underway for probably a decade or more. A lot new initiatives are currently planned. And then evaluating how well those are working and adjusting as needed and sort of adapting as you sort of go through this cycle of knowledge, action, and then learning from those actions. And so the space that I kind of occupy in this support for the conservation and restoration of coregonines is up here, sort of the resolve, the taxonomy of these ciscos is to understand the diversity a lot better using genetics and ecology. So why are we interested in understanding the diversity of fishes? Well, one of the principal concepts that underlies our research is that a diverse portfolio is going to stabilize your ecosystem. And this is directly pulled from an idea of portfolio theory that in a very colloquial sense, I think most of us are familiar with. So most people have heard of not putting all your eggs in a single basket, right? That's sort of the basic concept. But in the 1950s, this was applied by Markowitz to financial and economic perspectives from a statistical standpoint. And really what the underlying stats is that when you have a lot of independent factors, the variance of those independent factors averaged is going to be lower than the variance of a single component of that system. And so the way it was applied in economics was don't invest in a single stock because the volatility of that single stock is going to be really risky. So buy index funds, diversify across stocks and bonds and your REITs or something like that. And so modern day, it's quite standard for people to manage financial portfolios that are diversified so that you can stabilize your returns over time and not risk losing everything with one asset. And what we found is this applies extremely well to ecosystems. So there's a lot of foundational work out of Alaska in the salmon fisheries that was done on applying this type of economic theory to the salmonid fisheries. And so this is from a paper that Ray Hilborn et al. Put out back in 2003, where you can see 3 stocks that were contributing to the Bristol Bay salmon fishery. And so salmon fisheries, they run up rivers to spawn, so they are river-specific populations, but then they all enter into Bristol Bay and exist in Bristol Bay, and that's where they're often caught. So they were able to track from quite early on what the contributors are and what rivers they were coming from a century or more ago to today. And so if you look at the contributors in this example, you can see that there were two rivers, the Naknek and the Nushagak, that were really the predominant contributors to that Bristol Bay fishery for decades and decades and decades. So that gray and that white river population that were contributing. And then the Ekigig, very small contributions to the Bristol Bay fishery. And that continues all the way up until about 1970. And then in 1970, they had massive declines. They actually closed the fishery, which is that big collapse there at the bottom. I think it was open for like 1 or 2 days for salmon fishery in 1973. So you see this big drop there as they're trying to protect the fisheries. But then after the 70s, when the populations start coming back, they start seeing this massive shift in which population is contributing most to the fisheries. And so what you can see is that Ekigig population is suddenly now a really significant portion of that Bristol Bay fishery. So if they had not been able to maintain contributors from all of those, you might go, oh, well, it's not a big deal. It's a really small population. It's just a tiny proportion of our fishery. But if you look over time and in the face of great change, suddenly that little small contributor becomes a really, really valuable component to that. So what we found is in numerous ecosystems, this is a really good concept to apply. And so this is just another kind of example of tracking a single asset and its variance versus the average over time. So this is from Daniel Schindler et al. In 2015. And the only thing you really need to know from this sort of noisy plot here is that the small thinner lines, black to blue, are individual river populations of salmon contributing to a stock or a broader mixed stock that's being fished. And then the thick black line, average of the abundance of those. And so what you can see is there's a lot of variance in the individual populations around those. But overall, the stability is much look at all of those components as one. So this is one of the underlying concepts that's really driving our science in terms of trying to protect, restore the coregonine diversity that's left in the Great Lakes. So the other aspect of trying to catalog diversity so that we can use a portfolio approach to its management is really encapsulated in the challenge of understanding what the diversity is. So this is a beautiful figure, I think, from von Lanthen et al. Looking at alpine whitefish radiations, and it just shows you like the sheer number of phenotypic diversity that's found in all of the sort of northern hemisphere radiations of coregonines that occurred. And so the challenge of this has been because these radiations occurred so evolutionarily recently, they haven't had time to diversify in a way that standard tools can really help us distinguish. And so it's been a real challenge for scientists working in these fisheries to understand how much of the shared similarity between phenotypes across different lake systems is just plasticity and reaction to similar environments, or how much of it is actually heritable genetic diversity or shared descent between species. And so the other aspect of this problem is that even as adults, they're very difficult to tell apart in the Great Lakes. So here's a great example from Walter Keltz when he was describing diversity in Superior, where he's like, Kai closely resembles Hoyi, and at all times, you might confuse the two. So even as adult fishes, we are struggling to sometimes tell these things apart accurately. So having a tool like genetics to help us do that is really valuable as we're trying to catalog what diversity is out there. On the flip side, you have a very important component to the life history of these fishes where you have absolutely no clue what you're working with. So when you're looking at the egg stages, when you're looking at the larval stages of the fishes in the Coregonus artedi species complex, we have no characters that will tell them apart. So this is a really critical gap in our knowledge of, are they, what does it look like? Like, when are they starting to hatch out? Do we see succession and hatches out? What are they eating? What resources are they using? Where are they occupying space in different lakes? And we don't know that because previously we weren't able to identify whether we were looking at a cisco larva, a bloater larva, a kai larva, something else. And so having a tool that can really help us understand this critical early life stage where there's a lot of stochasticity and mortality is going to be very beneficial to understanding the diversity that we see and the changes in population structure we've seen over time. So normally, if you were a geneticist, you might tackle this problem with something like mitochondrial DNA. This is your classic barcoding, markers that you might use, like 16S or CO1 or Site B. And what we found is because these things are so evolutionarily young, that doesn't work. So this is just an example from a paper that shows that in clade A and clade B there, you see 3 different Coregonus species all just sort of intermixed, unable to be told apart with mitochondrial DNA sequencing. Okay, great. So that doesn't work. What about microsats? Microsats became king for quite a while. They were really helpful. You look at repeat regions that have more likelihood to mutate faster. And so that's been a very useful tool for people to use to try and distinguish things. Well, microsatellite DNA, not so great for these either. So we were able to start seeing really broad patterns. So this is a lot of work has been done historically in coregonines in North America through Laval University and Louis Bernochet's lab with Julie Turgeon back in the day. And so the work that they were able to distinguish is that they could find a western and eastern clade, essentially, that sort of spans the Great Lakes region. And so you can see on the tree there at the bottom right where those 2 western and eastern clades fall out. But species-wise, you're still looking at a lot of lack of clarity within the clades with microsatellite DNA. So that's an example of what do we have? We've got bloater, we've got short nose, and we've got Cisco there that are just found all over that tree within the Atlantic clades. So still not really good at distinguishing. But what we found and what I found when I started to my postdoc at University of Wisconsin-Stevens Point is that when we started applying higher powered methods that survey more of the genome, suddenly we were getting clarity. So I started working with applying rad sequencing data. This is a great figure here on the left from a student that I worked with at the University of Vermont for her master's degree, where she was looking at it. And you can start seeing we now can very clearly distinguish between the 3 common species in Lake Superior. Cisco, bloater, and Kai are very easy to distinguish once you start actually surveying much more of the genome. So rad sequencing samples, about 1% of the genome. We're now working with whole genome sequencing. So we're looking at, tens of millions of SNPs in some cases. And with that, again, we see very nice clarity. So that's something I started. That's something Kristen is taking forward and working with us on to better understand the diversity of that. And so now that we have these big tools that are really helpful for distinguishing species, how do we apply that in a functional way? And functional, I mean, rad sequencing is not super cheap. It's not horribly expensive. It's not super cheap. But one of the things it absolutely requires is high quality DNA because you are shearing the DNA. You need to have large fragments to start with to make sure you're getting the same loci across a number of samples. So the more degraded your DNA are, the more samples are just going to drop out by random chance. So you really need beautiful quality DNA, and it's moderately pricey. Whole genome sequencing, much pricier, much more expensive. So this isn't something that you can just deploy quickly and on the regular. So one of my goals, once we started being able to distinguish these, is let's mine these loci. Let's pull out the most informative loci into a small subset of loci that are, across the genome that are representative that can help us distinguish the species from each other and give us a tool for ID. So I developed a genotyping in thousands by sequencing panel, or a GTseq panel. And essentially what this was is approximately 500 of the most informative loci that we could pull and just massively multiplex into a single PCR. So we basically run a single PCR reaction. We get about 500 different regions. We're targeting an amplicon that's on average about 100 base pairs long, which means it can deal with degraded DNA a lot better. And then we just sequence that in-house on our MiSeq. And suddenly we are able to have a panel that can not only tell apart beautifully the 3 species that we designed it to tell apart and the lakes that they are found in, but now we are starting to see how well it adapts to other areas that it was not trained on. So we trained it on the RAD-seq data that could tell apart lakes, could tell apart the main species, but we found it cross-amplified on lake whitefish really well, cross-amplified on the round whitefish, totally different genus, and enough that we could actually take something designed for one purpose and now expand it out so that anytime we run into, say, a larva, we can tell you just with the panel, is it cisco, bloater, kai, lake whitefish, round whitefish, pygmy whitefish, what are we working with here? So now we suddenly have a tool that is a lot more cost effective. And so we have been applying this on instead of tens or hundreds of samples, we're now applying this on tens of thousands of samples. So our first sort of look at this was, how can we apply this to some of the larval collections that are in Newston tows across Lake Superior? So the maps here, some of our preliminary work, all the panel and this sort of pilot work is going to be up as a preprint, hopefully by the end of April. So it's coming out really soon. And now we can actually start having a tool that we can dive into to better understand the early life history of this species complex in the different spaces that it's occupying. So where are they found? When are they emerging? So this is just pie plots of where different species were found and collected in the Newston tows. On the right is a plot just looking at sort of like what was the length and the species collected between the 2 most common species we were getting, so cisco and artedi, where you are able to sort of combine data sets that are a similar cohort. You can also start looking at what are the growth rates between the two. How do those compare? And so that's where you see our cohort from like beginning of June to end of June was more geographically similar and more close to each other in time. We could start estimating growth rates or things like that using these data. So what I'm going to focus on for the rest of my talk is sort of 2 different projects that we've used the GT-seq panel on to help better understand diversity. So the first thing I want to talk to you about is a project we had running trying to describe the contemporary diversity of the cisco species complex in Lake Superior that had a very surprising outcome. So historically, back when there was a monograph put out, our best understanding of the diversity in Lake Superior were that there were 5 species that were present. You had the 3 common species, pretty decent sized populations remained, and then you had 2 rare species, blackfin and Xenithicus, and those are really periodically caught. You know, we might have over a 20-year period, we might have gotten 20 blackfin or something like that, so extremely rare. So we wanted to tackle this from a morphological and a genetic standpoint. So the goals are, let's catalog the diversity that's remaining in the lake. Let's look at how Keltz's original descriptions of the 6 species that were described from the lake, how do the fishes we catch match those descriptions? So it's the 5 to the left that were presumed to be present in some level in the lake, and then short nose as well. And then let's try and compare those 2 types of diversity to one another to try and better understand, are there characters that we can use to better identify these? And then because we actually originally had planned to do this as a RAD-seq based project, but because when we got the DNA, the samples were pretty bad quality, they'd been frozen, the DNA was really sheared, we're like, okay, we've got a panel. Let's see how the panel does on distinguishing diversity. So in Lake Superior, we've very consistently had these higher power approaches over the last 5 to 6 years be able to distinguish the main species, right? So left to right, that was the first work out of my postdoc there with RAD sequencing. Then it was the work I did with Hannah for RAD sequencing. The one on the right is, again, it's transcriptomic data from Moises Bernal while he was out in Trevor Krabenhoff's lab for his postdoc in Lake Superior. And so what we see is a nice cluster for cisco, a nice cluster for bloater, and a nice cluster for kai. This is what we've consistently seen with any kind of genetic approaches with this. Now, some of these we were specifically trying to include some other species. So you can see that in Moises's paper there on the right, they did include Xenithicus, but it was mostly kind of falling out with bloater. And then on our end, we had a handful of species we genotyped from blackfin and Xenithicus, and those either appeared to be misids as well, or fell out somewhere in between. No distinct clusters in the small numbers that had been run before. Well, in this case, we were surveying a lot of fish, about slightly over 600 fish that had been categorized morphologically as one of any one of those 6 Celts species. So when we looked at that, we were able to pull about just under 1,000 actual SNPs from the 500 loci that we target with the panel. And the color that you see on this PCA here is related to the morphological ID. And so if anybody is less familiar with PCAs, this is partitioning your variants across different axes, in this case in 2-dimensional space. So PC1 describes the most variants, PC2 the next most variants, and so on. And each point is an individual fish. So what we can see is something, we typically saw before, right? So everything that tended to be described as Cisco falls up here in this sort of cluster space. And then the vast majority of what we called bloater fell into this cluster here. And then, of course, all the Chi that we found is right over there in that teal color. But there's a lot of other stuff going on here, right? So there's a ton of short jaw in bloater, in that bloater cluster. There's a ton of short jaw sort of else. There's like one short jaw in the Cisco cluster. There's a bunch of short jaw over there near Chi. But all of the short nose, that purple, anything that was described as a short nose, which is supposed to be an extinct species, was falling out near Chi. So like, all right, let's explore other axes. When we explored other axes, suddenly, there's your Chi right there that it's overlapped with in PC1 and 2. But when you look at PC1 and 3, Chi clearly pulls away. And then we have this 4th genetic cluster. For the first time in any work we've done, we've now found a very, very clear 4th genetic cluster that, with the exception of a single fish, is comprised of all the things that we described as short nose, except for this one that appears to be sort of kind of a perfect F1 hybrid between sort of the bloater cluster and the short nose cluster. And so if we actually look at the statistics of this and we say, okay, k-means, please tell us how many clusters you would find without any sort of preliminary knowledge of what things were described, what would you find? It says there are 4 clusters in your data set. We also ran this in standard methods like structure clustering algorithms. Again, 4 clusters, it's finding 4 clusters. And so this is just a bar plot showing those structure results there. You can look at each line as an individual fish, and the label on the bottom shows like we grouped everything by how it was ID'd morphologically. So you can see that things that are morphologically described as artedi are mostly in that red cluster, hoi mostly in the green cluster, chi mostly in the teal cluster. You can see the mis-IDs happening there. And then you get to Nigripanus, blackfin, there's a small number of there. Anything but anything different, and like not a different thing. It's a lot of bloater. Looks like there's maybe a chi or 2 in there. But then you get to Reichardti, after our friend Jacob Reihard. You see that as a very distinct 4th cluster with very few exceptions there. Everything is 4th cluster, except that one, what looks like hybrid. And then, and then you get to Xenithicus, and again, it's a hodgepodge. It's a bunch of bloater, looks like it might have been called Xenithicus. There's a chi in there I can see, and then there's a handful of things that are probably short nose. So this is an incredible result that we've never seen before. So if we take these 4 genetic clusters and then we start trying to inform morphology and understand how they connect to one another, we can take a look at the most informative characters that associate with the genetic species that we might be finding. So in this case, you've got 2 more PCAs. These are colored now by genetic ID. but their position in these PCAs is based on the morphological characters that were measured. So this first one has all 38 characters that were considered as we evaluated these. And what you can see is things that are genetically Cisco are very clearly differentiated from all the deep water things. And then there's a lot of overlap. You can kind of see Kai pulling out a little bit, but there's a lot of overlap in deep water with all 38 characters. So we run an analysis to tell us what the most informative morphological characters are. We found that 3 characters defined 95% of the variance that separated these species from each other. So those are orbital length, the length of the 3 gill rakers nearest to like the arch inflection point there, and then the spacing of your gill rakers. So a lot of stuff really associated with probably a feeding niche and potentially the depth that they are found. So if we run the same kind of analysis we did with genetics, let's blindly take these morphological characters and say, how many clusters do we have? Well, our stats were telling us if you just look at these 3 morphological characters, again, bit circular, but there are 4 clusters there. And if we ask it to assign a species or a cluster to a group of fishes without having any idea what the genetic ID is, we find that there is a really high accuracy. I believe the accuracy was about 96, 97% accurate when you just use these 3 characters to predict a species. So the colors there on the bottom right PCA are the predicted clusters based on those 3 characters. The colors that are on the upper right PCA are based on how we genetically ID'd them. So you can see just from those 2 pictures how similar those outcomes were. So our understanding is a little different of what's in Lake Superior than what was thought to be there. We thought there were 3 common species and 2 rare species, but now we know, yes, there are 3 common species, but there is, at least to our best of our knowledge, 1 true rare species, and that is not blackfin or short jaw. It's rhyhurdi. It's short nose, a species that we thought was completely extinct. We don't know what the status of blackfin and Xenithicus are. We don't know if they ever truly existed or if they were always potentially mis-IDs. We are currently working on trying to understand that, but this work is actually out as a preprint and it's in revision right now for publication. And so hopefully all this will come out fairly soon over the next couple of months. But this panel has been really valuable. We thought we were just going to kind of look at diversity and morphological characters, and we stumbled across a species we thought was extinct is actually not extinct. So let's see, the next one I'm going to talk about in the last tale that I'm going to tell you about how we are using the GT-seq panel is to help describe the historic species diversity in Lake Erie, which has been completely, all Cisco's and any coregonines that existed there other than lake whitefish have been completely extirpated. And Lake Erie's history is a very unique story. So Walter Keltz himself, when he put together a report for the Fish Commission, had talked about the decline of these fishes very early on. So he said in 1925, the impossible, the incredible happened. The herring of Lake Erie suddenly and without warning gave out. Where in 1924 the fish could be caught by the ton, in 1925 only stray individuals could be found. As a fisheries biologist, I had always heard about overfishing, but I had never quite seen it in action quite so much. And so here's sort of the plot of how many fishes they were pulling out of the lake. The species of Cisco, the particular population of Cisco was a subspecies called albus, and it had sort of like big shoulders, which gave it a real wide body, which made it a lot more of a popular fish, commercial fish to fish. And so back in sort of the 1920 period, they were pulling a massive amount of Cisco out of the lake. And so they were maxing out, at about 35 million pounds in production. And so I always try and put this in terms of context. And so because we're a Department of Interior, American bison are a nice context to try and use. An adult male bison is huge. In 1 year, that's like taking 22,000 massive adult male bison out of the lake. Or, as is relevant to especially last night, that would be about just shy of 900,000 wolverines in a single year taken out of the lake, which might have approximated roughly the amount of people that were celebrating last night in Ann Arbor. So the scale of the fishery was incredible. And just as a quick sort of illustration of that, one of the common methods they used for pulling fishes out of Lake Erie were pound nets. And the scale of these pound nets was mind-blowing. So in the bottom right-hand corner there, you can see a standard pound net setup where it's just a very long line of netting that then goes into kind of this heart-shaped pin. So the fish would be swimming along the shoreline, hit one of these pound nets, and then divert along the pound net and get caught into that circular pin in a way that they couldn't get out. And so the length of a single one of these pound nets was probably about 300 meters or so. And these pound nets stretched up to 20 miles interconnected all along out from the shoreline in the western basin of Lake Erie. So they just, as they came in, they would undertake these big migrations west and east. And as they came back into the western basin, they were just slaughtered by these pound nets. And in fact, if anybody is continuing to do research here in the western basin, you know that a lot of the pilings from those pound nets are still navigational hazards today. So the sheer amount of fish that were brought in, were frozen. There were so many caught. A lot of times they would rot. A lot of times they were using them to feed like livestock and stuff like that. The amount of extraction was incredible. So how do we even start with something that's been totally extirpated? Normally one might be, interested in going to a museum, right? Let's look at what historically was there, what was caught. Let's find some of these specimens that are collected. But when it comes to genetics, museum specimens are generally off limits. That's because they've been typically preserved in formalin, which causes all sorts of structural problems with DNA. It also inhibits downstream reactions. So it is notoriously difficult to get any kind of DNA from a formalin-fixed specimen. However, we have a massive archive of scales that are retained at our center. So if you pull out one of these boxes from this archive, it's going to look like this. Inside, you've got a bunch of other smaller boxes that has a bunch of envelopes. This is the kind of envelope you might have here, and this is how they were aging fishes for decades. So as they were trying to develop cohorts and models of fishes, they would use scales to age. But at some point, scales became not the gold standard for doing aging. Otoliths hard parts became the gold standard for aging. And so a lot of these scale collections where they would just scrape a fish and wipe it off in a piece of paper and store it in an envelope, they just got stuck in closets because it wasn't the best science. It wasn't the most accurate way of aging a fish. So we don't want to use these anymore, right? So they got stuck in attics, they got stuck in sheds, basements. People have found them in very strange places. And for a long time, they were just ignored because they were there for aging, and that's not a good way of doing your aging science. But now that we are able to access very small amounts of DNA off of things, these are beautiful gold mines for us doing genetics work. And we can also gain a ton of information. So this is an example of a Cisco envelope that was collected in, let's see, Lake Erie from 1928 in October. Stillman Wright was the collector in this case. That was one of the scientists hired by Van Oosten when the laboratory was first set up at Michigan. And so he went out there and collected this. You could see that on there. And then we have a number stamped on this envelope that links it to a collection record. So we can actually go back to the record and find that. Those collection records also have coordinates. A lot of times they're going to be a little bit special to untangle those coordinates, but we can take things that are like Erie, Pennsylvania, 12 north by west, and turn that into an actual approximate coordinate for where that fish or those scales from that fish were collected. And so this is what you might see when you open an envelope, a bunch of fish scales that were scraped off of a fish. We can extract those. In this case, this is after extraction, a bunch of scales that we are returning to the collection because the scale itself doesn't go anywhere. And there's our DNA. It's very small. Just for context, this is good quality DNA from fresh samples. So that's 1928 samples from historic Lake Erie Cisco. We're very, very small fragments, but our GT-seq panel is targeting 100 base pair fragments. So if we can just get a few, enough fragments that will bind, then we can get this to work. And so what we wanted to look at is how did Erie potentially compare to the populations around it? So we had two contemporary populations we could pull into this analysis. The yellow is the Saugeen Bruce Peninsula in Huron. We also had some samples from Chameau Bay in Ontario. And then we included historical samples from Lake Erie and actually also some collections, historical collections from Saginaw Bay. That was also a really huge historic population that collapsed. Compliments of Paris Schofield during her master's work worked those up for us. Because Saginaw Bay and Lake Erie, because of the connections between the Detroit River system, maybe those are similar populations to each other. So let's take a look. So here's another PCA for you. This time we're looking at a 3D PCA. So right now it's oriented so you mostly see PC1 and PC2, the major sources of variance. And if you take a look at what we see here, our Lake Erie Cisco differentiate from everything else on that first component. So very, very distinct genotype from Lake Erie Cisco of those big sort of wide-bodied Cisco that once existed there. And then you can kind of see on that axis, you've got things like Lake Ontario, Lake Huron, and then even Saginaw Bay looks very different from contemporary Huron stuff, but not at all similar to the Lake Erie population. And we do see some things that look like there could be possible immigration occurring between Huron and Lake Erie as well. Things that look sort of in between the Saginaw genotype and the classic Lake Erie Cisco genotype that was found in all three locations. So there are periodic fishes caught, even though we say it's extirpated because there is no functional population there. There were 8 Cisco that were caught between 2010 and 2023 in Lake Erie, mostly in the Western basins. These are the dates and locations. Niagara Reef had, I think, 3 fishes, so those points are on top of each other. But we genotyped those with these population data, and they were all Lake Huron genotypes. So what we think is we're getting immigrants coming from Lake Huron and using that Western Basin habitat to potentially, in some cases, spawn on, in some cases just use for foraging because they were caught in times like April. Okay, so no remnants of these seem to exist in Lake Erie anymore. What about elsewhere? So we took some contemporary populations there. So again, you can see there's a Lachino population, the Bruce Saugeen Peninsula, also Perry Sound in this group. And there was a collection in Crystal Lake, Pennsylvania, which is this tiny, tiny, tiny inland lake squashed between a couple golf courses that was really similarly morphologically to the historic Lake Erie Cisco. And a bunch of scientists working at Pennsylvania Boat and Game and the USGS were like, there's some stocking records here that indicate they may have been stocked, but we don't know if they're stocked from Erie or if they're stocked from Ontario. So it's like, all right, well, let's take a look at these with the GT-seq panel since we have some data now to see, can we tell, were they stocked? Are they different? Are they totally drifted? What might we find? So, and this is just a good example of like how similar they look. So that the top right corner in that group of photos right there, that's a historic Lake Erie fish from the museum actually here. And then the bottom is a crystal lake fish. So you can see those big wide body types are just look like mirror images of each other. So here's all of our baseline populations, all of our references. And so we've kind of oriented it here a little differently. So axis 1 is actually up and down, so that y axis there. And axis 2 is on the sort of X associated axis. So those blue, those are our Erie Cisco. And then pink is Chameau Bay in Ontario. So somewhere along those, we think maybe we would see something fall out. So we put in the Crystal Lake Cisco, and I was not expecting something this amazingly beautiful and clean. Wow, right on top of our Lake Erie Cisco. So not only does it look morphologically like it, and there are some records that were pretty decent at saying we think it might have come from here. Now we have genetic data saying this is big. So we do actually see some evidence of drift. If we flip this and look at PC3, so coming out of Lake Erie, you see evidence of those genotypes starting to drift because they were stocked 100 years ago. They were stocked in, I think they're 1920 or so. So like this is just really exciting and really incredible because it was a very unique, at one point, this was the world's largest freshwater fishery. Not the United States, not North America, the world's. And we thought it was completely gone. And now what we found is what we call basically a bucket pop. They just randomly stocked a bunch of fish into a couple lakes, and we have some survivors in this tiny little private lake stuck between a couple of golf courses in inland Pennsylvania. So this is sort of our Lahontan cutthroat trout for people that are familiar with salmon work in the West Coast. The state fish of Nevada is Lahontan cutthroat trout. This was another species that it was well known, a huge important fishery, and they thought it had gone extinct because it was so overfished, and people loved it. It grew huge, 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 and it was gone for decades. And then somebody found, and I highlighted it because I just love the description, in a dinky stream, they found fishes that looked like juvenile ones, but they didn't get big. They pulled them out, they stocked them in a big lake again, and then suddenly they grew big. And after a while, they were able to pair the genetic work and found out it was a bucket pop. They were able to actually reclaim a species they thought was extinct. So now we have our own version of that in the Great Lakes. So that's the two tales that I'll show you about how we're using the GTZ panel. It's kind of our bread and butter these days. We're using it to survey historic diversity in other lakes, and we're building on it to get a neutral panel that we can use for monitoring stocks and hatchery stocks. We're using it to pre-inform a lot of our other work, a lot of stuff that Kristen is helping with, surveying whole genomes, looking at adaptation, and we're continuing to sort of build upon that with other avenues of research. But I know I'm getting close to my time, so that's all I have for you guys. All of this work is extremely collaborative. We can't get these collections without a lot of assistance from a lot of partners on both sides of the border and across a lot of our partnering agencies. And if we have some time, I'd be happy to take some questions.
Thank you, Amanda, for that amazing talk. So we do have a few minutes for questions. Margaret, I think, has a microphone that she'll bring around, so please use that so the online folks can hear. And then Margaret will also alert us if there's questions online. I had two quick questions. One, is that remnant population being reintroduced into Lake Erie? And yeah, so that one first, maybe.
Okay. There is a ton of interest in preserving the population. So two levels. If they could get it to a production size that they could start reintroducing, I think they would like to do that. The first level that especially the Pennsylvania Boat and Game people are really interested in doing is protecting it. And so that means not just trying to protect the lake, which is actually not a state managed lake, it's a private lake. It's trying to collect fishes and make sure they're not pulling out too many to affect the lake population and get them into hatchery settings so that at least it's in two places and they can start looking at how the hatchery production might be. But yes, they are very interested in doing that. And a lot of this work came out because they wanted to reestablish Cisco populations in Erie. And they have some work underway from New York side of things to pull Lake Huron genotypes in there. But everybody has got their eyes very strongly peeled towards this historic form.
have lots of other questions, but I should let others ask because I'll have lunch with you. Jesse.
Great talk, Amanda. Jesse Fine here at Gloral. So if you did reintroduce them, how do you know if they would thrive? Because they've been gone for a long time, and like, is there food for them? Who would they be competing with? Who's going to eat them, etc.? Because I'm guessing, we don't have the same lake that we did when they were last thriving.
Definitely not. And all extremely good questions. So I would say the easiest answer is we have no guarantees when we are trying to stock or reestablish anything at this point. What we do know is that the majority of what led to their decline was overfishing. It was human-driven exploitation that really wiped them out. So yes, they were impacted by lampreys. Yes, they were impacted by other invasives. But because one of the major reasons why they were driven to extirpation out of the lake and just extinction as a subform of Cisco was sort of a human-driven effect. And because of that, I would say that there's a fair chance that they could, if they were able to get a stable enough source in a hatchery going, they could potentially work on it. I think it would take a lot of time. We do have evidence of coregonines coming in from Lake Huron and spawning. So how long it would take for a population to really build up is unclear. So yeah, and with things like mussels and a lot of changes, yeah, we just don't know for sure until it's attempted. But efforts are underway to stock Lake Huron Cisco. And so understanding what type of return and how long that effort might take could sort of be benchmarked off of some of those efforts as well. But very, very good, relevant questions that are very difficult to answer.
Just a few questions about where they were found. So they were found in a private lake, and how large was it? How were they obviously they were reproducing there, and were the owners even aware of it, I guess?
That's a good question. And I have some of those answers and not others. Let me go back to the picture. So you can kind of roughly get a sense of it. I don't actually know the surface area of this lake, but it's sort of, I believe there's only like one or maybe two deep pools in this lake. And they were stocked 100 years ago, and that was it. And so it wasn't something that had to be repeated efforts or that they have records of them doing consistently over time. It was, a small effort 100 years ago, and they've survived. So at least in terms of persisting in this lake when a lot of Cisco populations and inland lakes are struggling is I think extremely promising. But they're currently working on a lot of acoustic surveys to try and understand how big the population is. And we're going to be doing some genetic work to try and compare the effective population size to other inland lakes that we've looked at to try and get a feel for how much could we extract to protect this population without impacting it. Yeah.
Oh, we have a question online. It's about where your new Cisco paper has been accepted. What paper or journal?
Well, if anybody's interested in Lake Erie stuff, I didn't put the link up on my paper. I should have done that. Joe Schmidt, one of our biologists out of the Sandusky office, has led a paper on the morphology side of things for Lake Erie. So if you just search Lake Erie Cisco and Joe Schmidt, his name's on this slide that's up right now, you can find that paper. My GT-seq panel paper and our preliminary larval work showing sort of proof of concept out of that and its amazing ability to cross-amplify is going to go up as a preprint. I am submitting in optimism to molecular ecology resources. It might get immediately booted, we'll see. But that means it will probably be a preprint on Authorea. And so if anybody's interested in knowing about that, they can email me and I'll let them know as soon as the preprint's up. And then the Lake Superior work is up as a preprint on bioRxiv. And that is in getting its final revision for CJFAS, so the Canadian Journal of Fisheries and Aquatic Sciences.
Okay, well, we should wrap up. Thanks everyone in the room and online for coming, and let's thank Amanda one more time.
Thank you for having me.